Hello, everyone. I'm Kelly Eversole, the Executive Director of the International Alliance for Phytobiomes Research, and I'm very pleased to be joined today by Sharon Huws from Queen's University in Belfast, who's going to talk to us about healthier red meat and reduced methane and how that actually will benefit the planet and the role of the microbiome in accomplishing that. But before we start with our webinar, I'd like to tell you a little bit about the Phytobiomes Alliance. We are an international alliance. We're a nonprofit consortium of industry, academic, and governmental scientists. We are sponsored by a number of organizations, both at the industry and governmental level, and as well as in academia. And we have everyone from small startup companies with no revenue to, of course, some of the larger companies that have been working in this space for many years. So what is a phytobiome? There's often a lot of confusion about that. A lot of people think it's just the plant microbiome, but in fact, it's really looking at the complex system of plant-based agriculture and how plants operate in a particular context and in a particular space. So it's the idea of a plant in a biome and all the interactions that occur with that. It also includes how it is influenced by management practices. And I know some of you may think, well, what in the world are we doing talking about animal microbiomes and how that works? Well, in fact, plants are a key component of the feed that goes into the livestock. And so that is something we're very interested in. And also we're very interested in this concept that we're going to hear from today about using algae and how that will work with other feed and plant-based feed, although I guess microalgae in some cases is considered like plant-like. So of course, this is one aspect that we want to pay attention to. In addition, in the Phytobiomes Alliance, we're really trying to increase our relationship with the livestock, poultry, and companion animal community, and really look at what we can do on the plant side to actually help with plant-based nutrition. So our vision within the Phytobiomes Alliance is that all farmers will have the ability to use predictive analytics to make the best decisions regarding management practices, the best varieties, and also the best genetics for a particular purpose, which might, excuse me, include animal feed, and to be able to do that in any given year. And I just want to mention that we will be having a multidisciplinary conference in September of next year. And we will be rolling out the call for abstracts before too long, excuse me, at the beginning, I believe at the beginning of 2022. And so we do encourage you to think about that and to think about the breadth of what we're trying to do and to bring all these different disciplines together. So just a reminder that our next webinar will be in November, and we'll be talking about boards and the giant extra chromosomal elements with the potential to augment methane oxidation. I'm really excited about the boards, whole concept of boards, and look forward to learning more about that. And you can sign up for that webinar now, and you can also get on our mailing list for the webinar series generally. So just to let you know, there on your dashboard, there's a questions button. Please put any questions that occur to you at any time during the webinar in the Q&A panel, and we will try to get to those at the end of our session. You can also monitor the chat panel, and you can see messages from the organizers, as well as you might, if something comes up and there's a reference to a website, we'll post that link on there. You can already download both this presentation that I've just given and Sharon's presentation. And also this webinar will be recorded and will be posted in the Phytobiomes Alliance YouTube channel. So without further ado, let me turn it over to Sharon Huws from Queen's University in Belfast. And she's going to talk to us about sea-derived microalgae and how that can lead to healthier red meat and reduced methane emissions. Sharon, thank you so much for being with us today, and I'm going to turn it over to you.
Thank you. Okay, so I'm absolutely delighted to be talking to you today about the use of microalgae to produce a healthier meat product and reduce methane emissions. And before I go on, I would like to say a very special thank you to the Phytobiomes Alliance for inviting me to give this talk. So the talk I'm going to give today is absolutely brand new. I have not given this talk outside the project before, so it's very new data. So the ruminant sector has many challenges. Some of the ones that I have noted there is obviously production. The world population size is increasing, and there is more strain on the food security of ruminant products. So production is a big target, but we have to produce in a more sustainable way, a way that actually reduces the environmental footprint of ruminants. And we also have to think about the health of the animal. Health and welfare are number one on the priority, and also the quality of the product that's being produced. So the healthiness in particular of the meat and milk for consumers. And this is the concept I really want to get across, is that for too long now, we have been studying each one of these parameters in solitude, and actually they are all interconnected. And we really need to look at interventions which address all of our global challenges. So I want to just introduce you to the rumen microbiome, because these are the most important microbes in terms of governing some of the targets that we have. So the rumen is one of 4 compartments within the GI tract of ruminants, and it's the most important one because it has the most microbes in there. It's the largest compartment, and it's also where all of the fermentation of feed occurs. So I have been fascinated by this ecosystem now for a long, long time, and that's really because of its complexity. It's one of the very rare microbiomes that contain bacteria, protozoa, fungi, and archaea, not to mention the bacteriophage as well. And you can see there the common densities that you would see. I do want to just point out that the archaea, although they're quite low in number, they are the ones responsible for the methane emissions coming from ruminants. So they might be not particularly dense, but they are very important. Excuse me. I want to just show you a video clip. So this is a rumen sample that we put underneath the microscope. The green blade you see there is a blade of grass, and the larger organisms moving around are protozoa, whereas the bacteria are tiny little spots. But you can see that it's particularly packed full of microbes. Okay, so the rumen has really evolved over the last millennia or so to break down carbohydrates, particularly cellulose, hemicelluloses, and also to break down starch products. That is what it does. And in the process of breaking down the hemicellulose, cellulose, etc., in the diet, volatile fatty acids are produced. You see them at the bottom there in the darker green. And these are mainly acetate, butyrate, propionate, although there are some intermediates like lactate and succinate that you see on top. And these volatile fatty acids actually provide the animal with the energy that they require. A kind of a side product of this fermentation is, of course, that it produces a lot of hydrogen. And when hydrogen is in excess, it goes down the methane pathway where you get CO2 and hydrogen combining to make methane. So I really want to just show you this because this is quite central really to looking at environmental impact and productivity. And also just to note, okay, you can see there that pyruvate to lactate actually utilizes 2 hydrogens, and then lactate to propionate another 2 hydrogens, and acetyl-CoA to butyrate utilizes 2. So there really is a kind of a hydrogen battle within the rumen as to whether it is used to make volatile fatty acids or whether it also is directed towards methane. And I've already mentioned this previously, but these put it into context in a visual way. You can see there what is happening in terms of world population growth. And then the other figure there on the right shows you that actually the demand for ruminant products isn't increasing across every country. It's actually increasing more across countries that previously didn't consume as much ruminant products, such as Asia. And the FAO predict that we will have to increase meat and dairy production, both of them by around 70% by 2050 in order to keep up with this demand. So I'd also like to highlight, that because of the fermentation that happens in the rumen, there is quite a correlation between methane production and productivity. So some of the data shows that if you look at the most productive animals, whether that's in terms of milk production or growth for meat, you normally find that they have that they emit less methane, because methane can result in about anything up to 12% in energy loss. So that energy is then not used for productivity in the animal. But I would like to caveat that though, because some of the dietary interventions to reduce methane don't necessarily result in higher productivity. Some of them do, but it's not always the case. Okay, so if we look at methane in particular, we know that we have an issue. We know that we are responsible for 18% of greenhouse gas emissions, mainly in the form of methane as a sector as a whole. And also we know that nitrogen, which is released in the urine of these animals, can be converted to nitrous oxide in soil. And of course, nitrous oxide is even more potent than methane. So we have to think about both of these greenhouse gases. And we are legally obliged to limit global warming. And this was via the Paris Agreement previously. Of course, we've got COP26 coming up as well, which will, I hope, give us an even shorter roadmap to achieving this. But what I would like to say from this slide as well is that, just to put it into context, if we were to reduce methane emissions from ruminants by 45%, this would actually reduce the global warming by 0.3 degrees. So we would see that reduction coming to play. And also, most of you probably are aware, the US and many countries in the EU have now put a climate pledge together ahead of COP26, in which they propose that we will need to reduce methane emissions by 30% by 2030. So the need to reduce methane emissions and nitrous oxide as well has never been so important for the sector. But having said that, we have to put this into perspective, because actually when you look at the cross-sectorial greenhouse gas emissions, it's quite clear that the fossil fuel industry have a big role to play as well. And they will also need to fight, well, they are already, to be very fair to these industries, they're looking for ways of reducing their carbon footprint. And also, we should also note that whilst methane has a higher global warming potential than carbon dioxide, it actually has a much shorter shelf life. So carbon dioxide remains in the atmosphere for 100 years, whereas methane remains there for 10 years. So we do need to consider that as well. But irrespective, as a agri-food sector, we really need to do something about our methane emissions. So I'm going to talk to you today about data that we have gathered within this big EU Horizon 2020 project, which is led by Professor Paul Cotter in Teagasc in Ireland. So Paul has previously given a talk for the Phytobiomes Alliance. So I guess I'm the follow-on who is going to talk to you specifically about our role within this project. And we have all of the linkages there as well for the, so you can find out more about the project as a whole. And we have a very active Twitter account. So the Master project stands for Microbiome Applications for Sustainable Food Systems Through Technologies and Enterprise. I have to say, I do not remember that off top of my head normally, as it doesn't roll off the tongue. So Master is what I'll call it from now on. So Master has partners from all over the world, whether they're universities, whether they're companies, etc. So there's quite a lot of people involved in that program. And Master takes a global approach to the development of microbiome. I'm sorry, I don't know why it's flicking there on its own. So it takes a global approach to the development of microbiome products, feeds and services, etc. I think this next slide is really quite nice because it tells you a little bit more that we are looking at microbiomes in the marine environment, in fish, in plants and soil, in animals. So that's the work package that I lead, which I will be talking to you about in a second. And also looking at waste and fermentation and food production processes right through to human health. So it has quite a circular approach and is based on using quite a lot of technology, using strains, etc., services and processes. I like to use this photograph. I like to think that I still look like that now, but I think that was probably about 10 years ago now, but I will continue to use it. So I'm going to talk to you now about Work Package 3, which I said that I lead, alongside my co-lead, David Ruiz-Llanes, who's based in CSIC in Spain. And in this particular work package, we are looking at ways of improving productivity, reducing the environmental impact of ruminants, and also ensuring that we are producing products which are actually healthy for the consumer. And we are also working across with partners in Work Package 5, which is led by Nicholas Agata and Chris Creevy, who's also based here in Queen's University, Belfast. And this work package is really centered around the technology and the computational infrastructure to analyze the data sets across all of the partners. So Work Package 5 is looking at standard operating procedures for sample collection and storage across the project. Also looking at standard operating procedures for not just 16S based diversity studies, but more so on shotgun metagenomic based sequences, even including isolation of MAGs, so these metagenomically assembled genomes that you can get out of your metagenomic sequencing. So we're really, really very fortunate because we have the people who are really at the cutting edge of these computational technologies that can take our data beyond what we can normally do. Okay, so these are the work packages within Work Package 3. Sorry, I'm just keeping an eye on time. The first one is looking at the host genomics in order to alter the rumen microbiome. So this is really looking at the development of marker traits in the ruminant that actually govern their rumen microbiome. In particular, looking at productivity and greenhouse gas emissions there. And that's largely led by Sinead Waters in Chagas and also Ilma Tapio in Lucca in Finland. And they are literally just increasing their numbers now of animals because you need substantial numbers of animals to make that a statistically robust approach. And then the second one we have there is looking at novel feeding technologies at different stages of life. So some of the work that's been completed there is looking at interventions right in early life where you can generally manipulate the microbiome a little bit more easily. And then last but not least, we have a work package looking at tools and mathematical models to predict animal phenotype. So largely around productivity and methane emissions. But specifically, the work I'm going to talk to you about now lies within this second objective here. So I'll introduce to you what we have completed so far within the master project. So we have been looking at microalgae supplementation in lamb diets. And we have been supplementing with microalgae which are rich in DHA, so long chain fatty acids, and monitoring the animals at the beginning before they receive the microalgae intervention. Then at 33 days post-intervention, and then a few days following this again, monitoring at slaughter. So what I should say is we don't do this alone in Queen's. We very much work with our Alliance colleagues in AFBI on this. So it's very much a partnership across Northern Ireland. So what we have done is we have introduced 3 levels of microalgae to the diet. We have a low level, which is 3 grams of DHA a day. Medium is 6 and high is 9 grams a day. And then some animals are on control diets with no DHA. And they were actually fed a silage basal diet as well. So we have 56 animals in total. 24 of these went through the methane chambers for measurement, and 32 of them were kept for the more productivity aspects. So the before and after treatment samples that we took were blood samples, rumen samples, urine, feces. We measured methane and all at those time points. And then when we got to slaughter, we were able to take more samples from the lower gastrointestinal tract. So we took small intestine, large intestine, and cecal samples. And at that point in time, we also split the meat into loins, shoulders, and legs. And we had a look at their fatty acid profiles at 2 and 7 days post slaughter. So this was the setup here, and you can see that each animal was in individual pens, so you could monitor exactly what they were eating, monitor their health more easily than if they were in mixed pens. Okay, so the data so far then. So in terms of what they were eating, so that's dry matter intake there, that tended to decrease a little bit in the high inclusion of microalgae. And I think that was really because we were at a slight tipping point there where the flavour perhaps wasn't quite as palatable. But when you look at the graph there with dry matter intake on it, although it's significant, it isn't an astounding drop, and it's more of a tendency rather than a p-value of below 0.05. So if we then look at digestibility of dry matter, organic matter, NDF and ADF, we see some very small changes there as well. But again, nothing particularly major, although some of them were just significant. And also what I should note before we look at some of these other productivity values here is that actually animal health was not affected in any shape or form. The animals were very happy and grew very well on the microalgae diets. So when we look at average daily gain there, we do see some slight differences with the low, medium and high, perhaps sometimes positive, on occasion slightly lower, and the same with relative growth. But these are really not overall major differences. But if we look at lamb growth, you can see there in the arrows denote where we have significant differences. And what you can see is with the red line, they are the animals that were fed a high inclusion of microalgae. And you see on occasion, they tended to have a slightly lower body weight at intervention days, whereas the low animals there tended to sometimes have a higher body weight than even the control animals. Okay, if we look at rumen pH, we did see a slight drop in rumen pH when the animals were fed medium or high inclusion levels of microalgae. But again, they were 7.1s, both of them, which is actually really very good. So nothing to note on the animal health side. When we look at methane emissions on a grams per day basis, we saw something like an 8% reduction in methane. So I would not scream and shout about this because it's not a major reduction, but it's still a reduction. And when we look at total volatile fatty acids, we also saw increases in these, particularly in the high inclusion of microalgae. If we then look at these separately, so if we look at the three main volatile fatty acids that are produced from the breakdown of complex carbohydrates, we can see that the acetate has a tendency to be reduced when the animals were fed medium and high levels of microalgae. And also to note, methane can be produced from that acetoclastic pathway as well. So we do want a decrease in acetate in the rumen, which we do see a tendency towards here. Very little differences on butyrate. And what I will point out on propionate is that actually we do have higher levels on medium and high inclusion rates, but they weren't significant. And just to remind you all, propionate is what we really want to be produced in the rumen, because it utilizes 4 hydrogens in its production and is a valuable energy source for the animal. Okay, if we now look at product quality and we look at the fatty acids within the loins in this instance, so the loins that we obtained post slaughter, what we can see there, the box highlighted in red shows you the DHA content of the loins. And if you remember, the microalgae we used was very DHA rich. And what you can see there with the medium and the high in particular is that the loins contain a substantially more DHA, and actually up to levels where they can be labeled as a source of omega-3. And if you look to your left, you'll see the omega-3 to omega-6 ratio as well, which is also substantially improved in terms of what we know is of benefit for human health. So they were really very good results. But then you have to start to ask yourself the question of, are they really beneficial? Because of course, when we go home, we don't eat the meat in a raw fashion, we cook it. So the first thing that we did was actually look at the effects of cooking and whether that diminished, some of the increases we were seeing in DHA in particular. And when we did this, we actually made patties or burgers. So we took the legs and the shoulders and we made burgers from those. And these were cooked before we looked at the fatty acid content. So again, I just want to highlight the red box at the top there, which looks at your DHA content. And you can see on the left-hand side, that's the fatty acid content of your raw burger. And on the right-hand side, that is the fatty acid content of your cooked burger. And you can see that actually cooking helps substantially, because if you look at the high inclusion level there, it has gone up because you've essentially taken the water out of the product through cooking. And we have tested these burgers that we made to make sure that their pathogen load was very low, and we found next to nothing on the product. So having found that, we decided to actually conduct a sensory panel to see whether what we had done in this experiment was palatable to us as consumers as well. Because quite often when you increase the long chain fatty acid content of meat products, they have a tendency to have a fishy taste, which is not desirable, of course. So we were meant to do this using a trained panel of experts, but then as COVID hit, we decided to do this as a home sensory panel. So we used staff and students, for example. So, and in a way, there's a lot of benefit to that because we are untrained consumers. So we will be able to say, you know, whether the resultant product is has any palatability effects. So we are just at the end of that now, so I cannot tell you the results. All I can say is that I was one of those on the sensory trial, and I personally did not notice much of a difference, but I don't represent all of the 80 or so people who were part of that, so we'll have to wait and see. Okay, so if we go on to what we've been doing on work package 5, excuse me a minute. So working particularly with Chris Creevy and Francesca Rubino on this, we have been sequencing some of our samples. So these would have been rumen fluid samples and buccal samples for before and after interventions. And then at slaughter, we also had the small and large intestine and the cecal samples. So we have sequenced these using the Illumina NovaSeq. So we have quite a depth of sequencing completed on those, and they have gone through all of their initial quality control, etc. And we're very, very happy with the quality of the sequencing completed. So what I'm going to show to you now is very new data. Actually, we only got this earlier this week, so this is all very new. So initially, okay, when we look at the bacteria and we compare control rumen fluid from control animals and those from those receiving medium levels of microalgae. So these are comparisons from the after treatment. And what we really see here, which is quite obvious, is right at the very top of the graph there, is the difference in the family Prevotalaceae. So the control diet seemed to have less Prevotalaceae in their rumen compared with those fed medium levels of microalgae. You see there where you see circles, that denotes that there is a significant difference. So there are also other differences there, but I just want to draw your attention initially to the largest difference, which is in the Prevotalaceae. When we then compare control to the high inclusion levels and look at the rumen fluid, what we see here again is some differences in the Prevotalaceae. It's not as great as it was for the medium inclusion level. And we also see some differences in some of the other families, but certainly we don't see the stark contrast that we saw with the medium inclusion level. And what I should have also said is that there were no differences in the archaea between control treatments and the high and the low microalgae inclusion. So I would say, you know, it's some of the reductions we saw in methane, although not great reductions, are probably really down to changes in the bacterial fractions in the rumen. So it goes to show that actually your reductions in environmental impact are not always because of reductions in methanogens, but they can also be due to the the fermentative capacity of your rumen microbes and how efficiently they're producing those volatile fatty acids, in particular propionate. And this was actually sent through to me this morning by Francesca Rubino, which is really, really very interesting data. Because if you look at the control samples versus samples, rumen samples, which have come from the medium treatment, the sequencing tells us that actually we have higher level of casezyme enzymes within the treated animals. So these are the enzymes that are responsible for the breakdown of carbohydrates, which is also really very interesting and perhaps also fits in with the slightly higher productivity that we see from the animals fed microalgae. And again, if we look at the control versus the high levels, it's not as stark as it is for the medium levels, but you do still see higher levels of some of the casezymes when the animals have been fed a high microalgae content, which is really quite interesting. So we still have a way to go, obviously, in analysing the data from that first experiment, but we have actually moved on to our second experiment within the master project as well. And in this, we are looking at the oil itself. So the previous experiment looked at freeze-dried microalgae, and I think a lot of the data that we've seen for the freeze-dried microalgae are due to the fact that the cell walls are intact. So they almost have a form of protection from breakdown from the rumen microbes, which means that some of the DHA has been able to go through without being transformed in any way to the meat itself. So we decided to look at the oil itself because the oil is actually easier to deal with in a practical sense. And we are working with our partners as well in Devonish who have a preference towards using the oil product. And ultimately within Master, we want to provide solutions and real societal impact, which is why we move on to look at these. So it's quite a similar experiment. We have matched the DHA levels that we used initially in the first experiment within this one. So the inclusion levels are based on the DHA that they would have been receiving in the first experiment. And again, it's the low, medium and high levels there versus controls. The difference in this particular study is that we actually have around 68 animals this time, so 24 for the chamber experiments. So if any of you work in the area of monitoring methane, you'll know that when you use a chamber, you cannot put all of the animals through because it's very time consuming. So 24 is the maximum that we can monitor for. Although with colleagues in AFBI, we have recently purchased some of the pack chambers, which allow you to monitor in a more high throughput way. And then we have 44, which are kept for the feed experiments. And it's exactly the same in terms of when we're going to take our samples. But we also decided this time as well, just to increase the length of the experiment. Because if you think about enhancing fatty acids in lamb, you have to think about the turnaround rate within the phospholipids in the muscle. And we believe that if with a longer inclusion, we might actually see some even better results. And this time as well, because the oil isn't protected in any shape or form, we expect to see some much better reductions in methane. The only thing to note about this, of course, is with oil previously, when you include it, you often get a shift towards the trans fatty acids. So we'll just have to see, because they are not as healthy for human consumption. So there's a balance within here, and we'll have to see what we get in terms of the all of 4 of those parameters that I noted at the beginning. So I believe I'm on time for a change. So I'm going to close just by saying that the free inclusion of freeze-dried microalgae at all of the levels had no detrimental effect on animal health or welfare, which is absolutely paramount. What we saw when microalgae was included, particularly at the medium and the high levels, was a significant increase in the long chain DHA content of meat and of the burgers that we made. And in particular, that was increased after cooking. And there was a very modest, I guess, reduction in methane emissions. But I think the most important thing to note there is the potential human health benefits of increasing DHA in the meat. So there were no major effects either on animal productivity. There were some on occasion, but nothing major. And you could look at this as a negative or a positive, because in fact we haven't affected productivity detrimentally, but in actual fact, you know, farmers get paid on production as well, so you would really want to have some productivity benefits, unless the meat is entering a niche market, if you like, whereby a consumer values the human health properties of the niche market. And the gastrointestinal tract microbiome analysis is very much ongoing. And the microalgae oil experiment now is over halfway through. So we'll see what we get. So this is what I would really say about the microalgae inclusion. We have a tick on animal health. We have a tick on product quality and human health attributes of the lamb that was produced. Production, well, there's a question mark there, because there was no detrimental effect, like I said, but also there was no enhancement. And we have very, very modest effects on the environment. So this is us here in happier times, I guess, when we actually could meet up together, and hopefully we can do this very soon. So I'd like to thank all of the master partners, but a special thank you obviously to Paul Cotter, who leads the project, and Mairead Coakley, who's the project manager, and all of those named afterwards are the local team, if you like, working on microalgae. But I would like to particularly mention Eva Lewis from Devonish, who are sponsoring this work. Okay, so I will end there and say thank you very much for listening.
Great. Thank you very much, Sharon. It's a very interesting presentation, and I have to say that your ruminant interconnected aspect is an image that fits very nicely with the whole phytobiomes concept, because it's really looking at that interconnected system. So I really appreciate that. So just to remind everyone, we are going to go through questions and answers now. And if we don't get to your questions during the live webinar, we will try to answer those subsequently to the webinar. All right, so the first question asks you to please elaborate a little bit on the microalgal selection process. What species were fed, etc.? So how did you do that?
I'm really sorry. I'm afraid I cannot disclose the microalgae species that we use, because that was very much chosen by our partners in Devonish, who are following it up in terms of a product, and they're using it already in another product, a poultry product, which is enhanced in omega-3. So the only thing I can say is that we chose a DHA rich product because DHA is so connected with human health. So I apologize that it's vague, but I know that Eva Lewis from Devonish is also probably on the line, and very glad that I'm not disclosing
Yeah.
that.
So I recognize that, and then one of the great things about a number of the EU projects is that they are very well integrated between the public and the private sector, and that actually helps to ensure that that the fundamental research ends up in commercialization in some streams in some way. So, but putting that aside, if you were to set up a new experiment, how would you go about selecting?
Okay. So there's quite a few different microalgae out there, and we are working with other species with partners in Portugal as well. I mean, some of them are EPA rich, some are DHA rich, and the long chain fatty acids seem to be reasonably equal in terms of human health attributes. So what we have done, actually, we do have some in vitro data which we're gathering now, where we're using a whole range of different species and also their extracted oil. So I guess in a way that turns the project upside down a little bit, because that's actually what you would normally do at the beginning, wouldn't it, to do that in vitro screen to see. But in actual fact, because we were working with Devonish, they already had a preference for this particular microalgae. But the in vitro data should actually open up other options as well.
All right, well, thank you for going in a little bit more detail as to what someone could use for their own projects. So the next question is, do you think the reduction in intake in the high treatment could explain the reduction in methane, or was that corrected for?
So when you correct for dry matter intake, it is true that you see a slightly less of an effect as well. So I think it's really important to be open about that, because certainly we're not screaming about the methane effect. It is actually quite quite low. So yes, it goes down even further in terms of a reduction when you correct for dry matter intake.
All right. So the next question, let me just get it to where I can see the whole thing, is one, were there any options to manipulate the hydrogen utilization in methanogens-free animals, since the high hydrogen, sorry, it's not all coming up, pressure could severely impair animal performance and health? And can we reprogram hydrogen usage?
Oh, okay. I'm just kind of thinking through that one and hoping that I've understood it correctly. So this is about improving hydrogen use as a whole. I wasn't quite sure, Kelly, at the beginning of what you read out. Could you possibly read that?
Sure, I'm happy to, and particularly given that I'm having trouble a little bit with, it's a long question, so it
Okay.
goes past my screen. So the question is, I wonder if there are any options to manipulate the hydrogen utilization in methanogens-free animals. Since the high hydrogen pressure could severely impair animal performance and health, for example, desulfovibrio species, pardon my pronunciation of that, and thus can we reprogram the hydrogen usage?
Okay, so that threw me initially because we don't have methanogen free animals because they are required, like the person says, to take the hydrogen away, because if you have too much hydrogen, you will have on occasion you do see some animal health issues. But I will answer that from the bacterial perspective. No, I think, you know, the bacteria angle and the utilization of hydrogen is equally as important as inhibiting the methane pathway and the methanogens. And I think we can do an awful lot on that. There's some recent publications showing that perhaps we can use things like direct fed microbials. So if you isolate some of the propionate producers, for example, in the rumen, then you can actually culture them and give them back to the animal, if you like, so that you're always maintaining high levels. So you're always encouraging that hydrogen to be redirected away towards propionate. And in actual fact, when we think about the lead products on the market at the minute, or have just been licensed maybe in certain countries, they actually reduce that methane pathway. But because it's not concomitant with a redirection of hydrogen via the bacterial VFA pathways, it means that you do not get a productive value. So there's no increase, say, in your growth of your animal or your milk being produced. So I think that there is a lot of value in that bacterial side as well, because it can add value to some of the compounds or some of the strategies that reduce methane, if that makes sense.
Yeah, it does to me. And if it does not make sense to the person who answered the question or asked the question, please put another one in. And if we can't get to it during the live, we'll answer it subsequently. And maybe that's a better way of answering a
Yes.
question, a
Yeah.
question like that. In the lamb's experiment with DHA, are there any correlations between methanogens abundance and unaffected methane emission?
Is this the first experiment, Kelly, or the second, do you think?
It probably, just in terms of when it was asked, maybe the second experiment.
We don't know yet with the second one. We are just approaching the after treatment methane capture, so we haven't got right to the end yet. So and we certainly haven't sequenced any of the microbial samples yet.
Okay, so continuing on with this particular series of questions, what is your opinion about DHA manipulation properties? Do you believe that DHAs act against bacteria and archaea membranes per se, or inhibit or disrupt bacterial biofilm?
So I think if you talk about the microalgae itself, the freeze-dried microalgae, which was high in DHA, I think that almost acts as if it was a protected oil, because the cell wall of that microalgae gives it protection. And actually some of Rui Bess's work in Portugal also shows that as well. So what I mean by that is that the microalgae can go through the rumen and largely not be too affected by the microbes within there. But we might be seeing an increase in some of the casezymes present. because they are trying their best to break down the cell walls of the microalgae. But in essence, they have some form of protection. In terms of the oil is a completely different story, because the oil, it just gets out there and it's not protected in any form. So, and because DHA has a lot of double bonds, it makes it very kinked in structure. And that kinked structure means that it's going to be more antimicrobial, and it can penetrate the cell wall of the bacteria, and highly likely the methanogens as well. And that is why I expect a much larger response on the methane side when it comes to the oil. I think the balance there is, as I said, we'll have to see what the trans fats look like as well, because we have to bear in mind that whole picture of everything that we're doing.
Right. So how were the microalgae produced, and how scalable do you believe the production would be?
I think our industrial partners in Devonish are better placed to maybe answer this. But just to answer, you know, they have a production line essentially of the oil at present. So they are also they are actually using the oil to make omega-3 enhanced chicken, which is sold in particular outlets. So it is scalable from that point of view.
All right, well, we're running out of time, but and just to remind everyone, we will get your questions answered, and they will be posted on the website or on the or the YouTube channel, along with the video. But I did want to ask you, I know you have a new project or involved in a new project that is integrating crop and ruminant livestock systems, something called the Integrity Project. You know, and that one is really near and dear to what we're looking at from the Phytobiomes perspective. Can you tell me a little bit about that project? I know it just got funded, so you may not have a lot of information yet.
Yeah, absolutely. That was really exciting news that came through last week. So it's a project which, from our perspective, we will be looking at multi-species swards for increasing productivity and reducing methane. And I find that really exciting because it provides a very practical approach. Of course, it's maybe not an approach that can be used globally. It depends on your soils and what you can grow, for example. But it does, and particularly on the island of Ireland here, it's being used a lot by the farmers. But I think, Kelly, you might agree with this. The only challenge at the minute is that multi-species swards are being used, but the combinations, I would say, haven't been looked into in terms of getting the best ones that have an additive effect, if you like. And also, you know, multi-species swards do make me smile, because it's a wholesome story, because you also increase your biodiversity, and the root systems as well are much better to be able to cope with extreme weather events. So what we will do, Kelly, is look at in vitro at various different swards on their own, in combination, and just have a look at their effects initially on volatile fatty acids and methane before we then move on to an actual lamb study, where we have some of what we deem to be the best swards on the ground. And again, I have to say that's very much with our alliance partners in AFBI. So
Yeah,
very
it's a really exciting project. So we look
yeah,
forward to hearing about the results of that further down the road.
yeah.
So again, Sharon, I want to thank you for spending some time with us today, and thank all of our participants for joining in. We look forward to seeing everyone on our next webinar, and hope that we see you at the Phytobiomes Conference next year in Denver, Colorado. So with that, Have a wonderful day, and we'll see you next time. Thank you.
Thank you.